I'm going to be discussing a little bit about the sirolimus and the Olimus family of drugs for applications for inhibition of stenosis in arteriovenous access. Those are my conflicts of interest. Now, angioplasty outcomes. You are hearing a lot. This is our common practice. It is quite routine to dilate the stenosis in the fistula. And you can see here the outcomes, quite poor patency results. Fistula do better than synthetic grafts, but still, this is some large series being published a few years ago. And you may note that patency outcomes are well below 50% at the 1-year mark. Now, why is that? Because stenosis in the arteriovenous circuits is not like in the arterial networks. We've got excessive, exacerbated endothelial dysfunction because the combination of oxidative stress and inflammation, and we get quite often what we call the negative constrictive remodeling, where the vessels tend to shrink, then dilate as flow gets higher. Another very important point is that when we discuss about the venous neointima, this is what we mostly know is composed by the myointima. So we get what we now call fibroblasts, and those are not like smooth muscles. Those are because they tend to express desmin instead of the mentin. And have a look here. This is some seminal work from Dr. Prabir. And the thinking is that we get fibroblasts from the adventitia, and they translocate into the media and into the intima, and then they start converting to smooth muscles like cells. But they do not express desmin, but they instead vimentin. So those are quite different types of cells. Another point to remember when we're discussing and thinking about the application of drug-coated balloons is that we get different types of stenosis and stenosis. We get what we call the constrictive type on the top left. We get the proliferative type on the top right, and then we also get the mixed type. So we don't always get this myointimal thickening, and this is something we might have to check and discuss more in the future. Now, there are many different ways to try to battle this and inhibit stenosis. You have heard a lot about the drug-coated, paclitaxel-coated balloon studies yesterday. I'm going to be discussing about something a little bit alternative. This is the way paclitaxel and sirolimus works. This is well known from the coronaries. It has been already translated to applications in the femoropopliteal segment. Basically, those are completely different types of medications. Paclitaxel is cytotoxic. It inhibits the cells in the M phase, in the mitotic phase, whereas sirolimus is cytostatic, and it inhibits the cells in the GO phase. Now, when we discuss about sirolimus, we have to remember that this is a very large family of medications. We have the sirolimus, we have everolimus, temsirolimus, zotarolimus, etc. So basically, what we call the rapamycin analogs or the rapalogs. Now, all of them work through the mammalian TOR receptor, and we get those mTORC1 and mTORC2 cellular complexes, and basically they inhibit cell proliferation, and they may induce cell apoptosis and cell cycle arrest. And remember, those are cytostatic medications, not cytotoxic medications like paclitaxel. What's the rationale for application of drug-coated balloons in hemodialysis? We have enough animal data from the past. There have been a number of studies on animal models of arteriovenous communications, and they have tested and shown a potential efficacy of both paclitaxel and sirolimus. The main differences in terms of the pharmacokinetics of paclitaxel and sirolimus is that the margin of safety, so the space, the difference between the dose of safety and the dose of toxicity is 10,000 times higher in the case of sirolimus compared to paclitaxel. On the left-hand side, you can see sirolimus. On the right-hand side, you can see paclitaxel. So basically, both medications have what we call the level of efficacy, which is at least 1 nanogram per milligram tissue. And on the top, you can see the threshold of safety. So approximately up to 10 milligrams in the case of sirolimus, and no more than 100 in the case of paclitaxel. Now, again, This is the first animal study with an application of a sirolimus eluting stent in the animals. Basically, they did a surgical synthetic graft arteriovenous communication between the carotid artery and the jugular vein, and you may note that at the venous anastomotic site, a sirolimus eluting stent was applied, and they have shown very nicely a significant inhibition of stenosis and better angiographic results up to at 28 days with the application of sirolimus eluting stent. Now, what does the evidence actually show with the application of paclitaxel in the drug-coated balloon studies in the fistulas? And we discussed about that yesterday. I'm highlighting here the three major multicenter studies, the IMPACT AV, the PAVE, and the Eutonics, the highest caliber multicenter studies published so far. There was a significant advantage in the case of the IMPACT AV, which is a high-dose paclitaxel-coated balloon, but out to 2 years, that was not the case in the case of the 2 microgram Eutonics studies. Now, it may have to do with the dose, it may have to do with different pharmacokinetics, the heterogeneity. It has been discussed yesterday, and the jury is still out. I will also note at the bottom, and this is from the FDA submission, that there were significantly higher rates of infections and pneumonias in the paclitaxel-coated arm in the case of the IMPACT AV study, and again, this is under investigation. So there are some safety concerns, and sirolimus-coated balloons may be an alternative. So remember, wider therapeutic margin and also different mode of action and diffusion, because sirolimus tends to spread out more into the adventitia compared to the intima and the media. Is there some positive evidence? Yes, there is in PAD. This is the rate lumen loss when we compare paclitaxel and sirolimus in PAD, and there seems to be similar benefit, at least in the femoropopliteal segment. What's happening in the fistulas? We have already 2 randomized multicenter controlled studies running, one with the Magic Touch balloon, 1.27 microgram per millimeter square, one with a solution balloon, different mode of action, different excipient, but both of those studies have already accumulated more than 2/3 of their recruitment target. So it's going to be quite an interesting future to see what's going to be announced in the immediate future. So in conclusion, sirolimus and the Olimus family may be an alternative to paclitaxel. They have shown efficacy in the coronary artery space and seem to be promising in the peripheral vascular space, and we are pending randomized studies in the arteriovenous fistula space. Thank you very much.